The coronavirus is attacking our nation. We will defeat it by implementing social distancing. That means we will no longer have classes in class. We will administer them online. We will defeat the coronavirus one lecture at a time with science. So this is a general lecture on Bt resistance and other resistance mechanisms in insects. Okay, so there's different types of resistance. There's many more types than just if you think of like classical antibiotic resistance mechanisms. If you're thinking about insects and plants, it's a lot more complicated things. It goes beyond the cellular level. So let's talk about the different types of resistance. Behavioral resistance. Let me shut these lights off. Whoa, whoa, whoa! Hang on, we're still good. I got hooked up. What would be an example of behavioral resistance?
They avoid the field. That's obesity.
Okay, if you didn't hear that, he said the insects learn to avoid the field. What would be go into that in more detail? Like how would they learn to avoid the field? Right, so there'd be some kind of like selection. But like what would convey that? Would that be like a brain thing, or would that be like a mutation, or like what do you think that would be?
Like pheromones?
Okay, so let's dig into that. So let's say pheromones. What would be emitting the pheromone?
The dying ones?
The plant. You mean the plant?
Oh, no. But yeah, okay, now I mean plant.
Okay, I was imagining like you have a plant. The plant is emitting these pheromones. And the insect is going to want to respond to these pheromones, right? And you're saying maybe there's a case where the insect then learns to fly away rather than fly towards this particular pheromone. That would be like you could get a mutation in whatever receptor was the receptor for that particular pheromone X. That would be an example of that. And that could be like the cause of that could be could lead to behavioral resistance. But it could also be just like a brain thing. You might not even need a mutation. It's kind of what I'm getting at, is that like insects have brains, they can learn. Some insects can learn, like bees. Bees have memories and they come back and they tell people things where to go. So like there is a scenario where maybe an insect it's possible that they could learn. And so maybe you're creating like different synapses that could lead to like behavioral resistance. Let's think about more behavioral resistance mechanisms. So let's go away from BT. Let's think about sterile insect technique. So sterile insect technique is when you have a bunch of males that are sterile. Let's say you have like a huge concentration of those and you release those into the wild. They mate with females and they produce sterile offspring or dead embryos. So we're going to talk about this more. But like a behavioral resistance to this would be, in this case, you're releasing male fly. It's a male fly that's sterile. And you have a female, okay? And the female has to mate with that male in order for this to work. And there might be something about this male. Maybe it smells different, going back to pheromones. Maybe it behaves a little bit different. Maybe there's something different. And this female can know that that's a male that she does not want to mate with. So this would be called like mate discrimination. Where the female knows that's a bad male. I don't want to mate with that one. I want to mate with that wild type guy right there who is fertile. And then so she goes and mates with him instead of him. That would be another case of like behavioral resistance. Okay. So it might be like a mutation, or it might be something neurological that changes in the brain synapses. It could be like a learned behavior, but behavior, you get the point. It's a behavior. It's not necessarily talking about a mutation per se. So that's behavioral resistance. Let's go to penetration. This is kind of a more general lecture on kind of like all types of resistance. And some of these might be pertinent to BT, and we'll talk about the ones that are, and some of these might be a little bit more pertinent to other things. Let's talk about penetration. So what would that be? What do you think that means? Think about insecticides.
Oh, how the chemical gets into their body.
Yeah, exactly. So the chemical has to get into the body. And insects are special how? Yes, they do. But they also have a shell. They have an exoskeleton. So their hard structures are on the outside rather than us, which is on the inside. And they also have a cuticle. And it's just like an egg. Just like an egg, where in the egg you have like an exoskeleton, which is like the chorion, and then there's also like a wax layer. There's a wax layer in the cuticle of the insect. And the wax layer has the same functions as in the egg. It prevents water from leaving, but it also prevents chemicals from penetrating into the body of the insect. And the cuticle also prevents chemicals from penetrating into the body of the insect. So in a case of penetration resistance, you could have, and this is not so relevant to BT because they imbibe and eat BT. You could get a scenario where if you spray a field with some chemical X and the insect, and before the chemical would passively diffuse right across let's say this is the cuticle. It would passively diffuse right across the cuticle. You could get a mutation in the structural organization of that cuticle because it's made of proteins that are cross-linked. It's also made of sugars that are cross-linked, and then there's a wax layer, and that wax layer has to be synthesized by enzymes. So you could get a mutation in any of these processes that might change the structure of the surface in a way that no longer allows that chemical to go through. So that would be like penetration resistance. So penetration can also be, that's at the body level, could also be at the cellular level. So this is relevant to BT, where BT, the Cry Toxins, have to get into the midgut, they have to get solubilized, and then they have to pass, or basically get into the membranes of the midgut has this epithelial layer of cells, and then up top here's the basal lamina. And so if this is the midgut, here's like the inside, the hemolymph, the body of the insect. Here's the exoskeleton. The BT has to get in right in here. It's got to get into that membrane. And there could be a mutation where the Cry Toxins are no longer able to penetrate into that membrane. Okay, so that could be a case of, I guess, in somewhat penetration resistance for BT. Okay, let's talk about metabolic resistance.
So
Yeah.
whenever you spray like a wasp with the
Like a
insect-
raid or something?
Yeah, raid. It's penetrating the wax layer on its body, or is it accidentally getting some of its mouth?
I'd have to look at what chemicals are in those sprays. A lot of those things will just straight up pass right through the exoskeleton, right through the wax layer, and they're usually neurotransmitter inhibitors. So this is the case with DDT and carbamates, and a lot of the insecticides are basically neurotransmitter inhibitors. But I don't know what the rate is. I don't know. I'd have to look at the chemical formula, and it might be a bunch of these things. So I'm not sure, because basically what I'm saying is it's going to be pesticide specific, whatever's happening. Good question. Metabolic resistance. What do you think that would be?
They can digest and get rid of the...
Yeah, pretty much. Digest is kind of like a word that you use for eating. So digest isn't quite the right word, but I know what you're saying. Kind of like degrade. Yeah, get rid of the chemical. And most of the metabolic resistance is actually really not degradation. A lot of it is in transporters. So each cell, right, if you have the cell, there's a membrane, there'll be these transporters that can put things in and out. And a lot of these times, these transporters there's detoxification enzymes, which basically pump bad stuff out of the cell. So a lot of the metabolic resistance will be related to those things, but you could also get enzymes that might degrade the chemical and turn it into basically a different chemical. It might cut off some group or something like that. And I'm just waiting if I want to go into this or go into the next thing. We'll use this to segue into the next thing, regulation. A lot of the metabolic resistance is related to regulatory resistance. I don't know if you'd call it regulatory, but regulation. When I say regulation, what do you think of?
Turning genes on and off.
Yeah, exactly. Turning genes on and off. So a lot of like metabolic resistance is like you just need a little bit more of a particular transporter that will detoxify your cell. So maybe you just get a mutation in the promoter that then changes the transcription factor that binds to it, and then it's in a state of being like perpetually on. Or maybe you're now expressing it at like 10 times what it was expressing before. So that's kind of like what you a lot of times see in the metabolic resistance, is it's conveyed by like regulatory mutations. Or you could get a mutation in a transcription factor protein. So if there's actually a gene that codes for a transcription factor X. So in this case, it could be, what's the lac operator protein regulator? What's it called? You remember? I think it's LAC-L, which is a protein that binds to lactose. But there's a transcription factor, right? And that transcription factor actually binds the lac operator sequence. Lac operator, let's say it's right here. Okay. And you could get an actual mutation in the gene that encodes that transcription factor right there. And that might change its affinity for its particular consensus sequence, or it might change its tightness to the binding. And so, or it might make it a little bit looser, so it's more likely to fall off. So things like that, mutations like that will change the regulation of genes, and that can change a whole metabolism of an insect. So there's a really interesting phenomenon where, let's say, in talking about metabolic resistance in insects, where let's say you have two pesticides. Let's say you have pesticide 1 and you have pesticide 2, and you treat some bugs with pesticide 1 and they become resistant. And then you treat them with pesticide 2 and they're also resistant to pesticide 2, but pesticide 2 is not the same thing as pesticide 1. They have different targets. How do you explain that? They're different. How do you explain this concept?
Have they never been exposed to the second one before?
Never been exposed to the second one before. How could you rationalize that?
Do they operate off the same mechanism?
No, I'm telling you it's different. Their targets are different. What?
Different targets.
Yeah, different targets. But it's resistant to both. How?
The factors which make it resistant to, like, enable it to destroy the first one.
Yeah, you're close. Finish that thought.
Okay, it's the enzymes that it upregulated in order to take care of that first one, then the enzymes are also capable of dealing with the second one.
Yes, exactly. So there's kind of like general resistance, and there's kind of like very, very specific resistance. So one of the specific resistances is like target site. So target site would be like if the BT, the Cry protein, the very, very specific receptor that it grabs, if there was a mutation in that particular receptor, you'd only get resistance to that Cry protein. That wouldn't convey resistance to, say, some insecticide. So that would be a very, very specific mechanism of resistance. But there's general mechanisms of resistance like the metabolic, which is like you're basically just kind of like changing the detoxification. It's like if humans upregulate their liver, maybe they're good at detoxifying ethanol, but they're also good at detoxing methanol. You know what I'm saying? Like if you're a heavy drinker, maybe you're also really good at detoxifying yourself if you accidentally drink methanol. Like it's kind of like the same thing. Like it's a metabolic thing. If you upregulate genes that are responsible for removing kind of like toxic chemicals in general, you might be able to induce resistance to multiple insecticides by just treating with one. And this is actually a problem that does happen. So you will see this. So there's kind of like general resistance mechanisms and kind of specific mechanisms. And you kind of just think in your head, which one of this is this likely to be? And so if it's kind of like you could imagine like if it was a transcription factor. So the LAC-L is kind of a bad example because the LAC-L is a very, very specific transcription factor that binds a very, very specific site, which is the LAC operator. Okay. And this is, of course, in a bacteria, not in an insect. But here's a question. Are all transcription factors that specific?
No.
No. There's some transcription factors that are kind of very general, that kind of bind a lot of different DNA sequences. So you could imagine if you had an upregulation of a particular transcription factor, that transcription factor might cause an upregulation. If you had a mutation, I'd say, if you had a mutation in transcription factor 1 right there, and that mutation causes it to have like high super functionality, it turns a bunch of things on. That's going to turn on like a whole cascade of like detoxification enzymes if it's in like a detoxification pathway. And you'll turn on a whole bunch of genes. And so that's maybe how you can get resistance to like a whole bunch of different like stress components by just a single mutation in a particular transcription factor. Does that make sense? Okay. Target site. Target site. Okay, so this is very common, again, when you think of like neurotransmitter type insecticides. So let's say DDT, I think this is correct. DDT inhibits or blocks sodium channels. You could going off my basal reptilian brain here knowledge. I think this is correct. But there's channels, right? And you know this from just neuron biology. Like there's channels in cell membranes, okay? And their function is to maintain the homeostasis of the particular ion concentrations in the cell, right? Like there's calcium channels, there's sodium channels, there's chlorine channels, magnesium channels. There's all these different types of channels, okay? And there's channels that are important for sodium in the brain, right? Because the axon potential, it propagates through an influx and outflux of sodium and potassium ions, okay? And I think DDT basically is a chemical. I won't be able to draw out the structure properly, but let's say here's the structure of DDT. It binds to these potassium or sodium channels. To correct a mistake here, it opens the channels, causing them to fire spontaneously. Okay? And so a target site mutation, what do you think that would be? A student inaudibly says here, a change in protein structure. Yeah, right, exactly. So this structure, this channel, is going to be a protein that's encoded by a gene, and you could make a mutation in that gene. This chemical is going to have some kind of a hydrophilic, hydrophobic, ionic interaction, right, with this protein, and that's going to be conveyed by the amino acid sequences in that protein. And all you might do is you might change a single amino acid, and then maybe that disrupts this interaction right there, and then this DDT chemical can no longer bind. That is target site resistance. And this is related to BT. We'll talk about specific mechanisms of target site resistance in BT. But that's an example of target site. Okay, last kind of type to talk about. Duplication. What would that be? What do you think that would be? Copy number of what? Right, that concept is the same. I don't know if it's specifically correct with DDT, but that thing you're thinking is exactly right. So we have chromosomes, and I guess, I don't know if this is true, but we're diploid. Most insects, I don't know if it's true that I say most, but a lot of insects are diploid. So there's 2 copies of each gene. Okay, and we talked about how maybe if you upregulate a particular gene with a transcription factor, you could get resistance, but you might not need to upregulate the gene. You might just actually make more copies of the gene. How might that happen? How could you make more copies of a gene? That's going to be making more copies of the protein. That's going to be regulatory. But I'm talking about the actual ORF, the DNA sequence. How could you make more copies of the DNA sequence? You guys should know this. I've taught it.
Like you just, we talked about it in the spider stuff. They just add repeating.
Well, that was like a cloning thing. So they had like restriction sites that kind of did something funky and kind of like made like a functional unit like this. I'm thinking more of like what genes are capable of copying and pasting. Yeah. So like there could be a transposon in the region. Let's say you got transposase here. Let's say its sites are here. Remember, I talked about this the other day. Let's say you got another copy of it, a second copy of that transposase, and let's say you got detox enzyme 1 right here. This transposase, okay, it might get made, might grab here and cut here and cut here, and it might take this whole segment, pop it onto a different chromosome that has this enzyme already there, okay? Pop it into a new chromosome, and now you have 2 of these detoxification enzymes on one chromosome, and that chromosome could be selected for in next generations until it becomes homozygous, and then all of a sudden you got 4 copies of that gene in your cell, not just 2. Does that make sense? So that could be one mechanism. Yeah, so like that's called those are retrotransposons. So transposons can only cut paste. They don't actually copy. Retrotransposons actually copy paste. So what they do is if they have retrotransposons usually have 2 genes. There's usually an integrase and a reverse transcriptase. Reverse transcriptase breaks the central dogma of biology in that it can take messenger RNA and it can rewrite it into complementary DNA. And then the function of the integrase is to take that cDNA and integrate it back into the genome. So the reverse transcriptase encodes a copy function and the integrase encodes the paste function. And they just like transposons, they have their own kind of like sites. They're called long terminal repeats on the left and the right. And they recognize this, and then they kind of tend to copy the whole thing, and then they paste this whole thing. But you can get errors where the copy mechanism goes beyond, okay? And maybe you have detox enzyme right here, and it copies the detox enzyme, and then it pastes that detox enzyme into the chromosome, and now you have multiple copies. That happens frequently.
Is it like a transposon that the insertion is random?
Okay, so at this point I went into a long tangent that did not need to be in this lecture, answering the student's question. Essentially, the point you want to know is that duplications on chromosomes can happen, and they can happen through retro elements, okay? And that can convey insecticide resistance. Okay, let's talk about actual Cry Toxin Resistance. Okay, so Cry, let's draw. Let's draw Cry Toxin in the gut. So this is in the gut of an insect gut. Okay, the first thing that the Cry toxins interact with is a protein called cadherin. I'm spelling this wrong. Cadherin. Okay, so I talked about there's this like layer of cells called the epithelia. When you hear epithelium, what does that usually mean? Have you guys had like animal physiology or anything like that?
Not skin, but a membrane layer.
Yeah, it's usually like the first layer of cells in some particular context. Like in the gut, the first layer of cells that blocks the inside from the outside is the epithelia. Okay, so whenever you see epithelia, you kind of think barrier between inside and outside in a cellular form. Okay, and each of these cells, do you think that the cells want to allow things to pass through between them?
Yes.
Well, in specific context, but I mean like everything. Do they want everything to just like flow through? No. So there's actually like junctions here that hold these cells together, like that enforce kind of like the structure, okay? And these junctions are called adherens junctions.
So it's not cadmium?
Hold on.
Oh.
Adherens junctions are the structure. They're made of proteins. What do you think the proteins they're made of are called? Cadherens. So the proteins that form these like structural things are called cadherin. And that's part of it. This is part of what's called, in a broader sense, the extracellular matrix. And there's lots of proteins that make up the extracellular matrix. Okay? But adherens junctions are part of that, and they're made of these proteins called cadherens. Okay, but the point is that they're kind of like on the outside of the cell. There's something sticking outside of the cell that's on the outside. If you are a toxin or a microbe and you're kind of like feeling around in the dark, the first thing that you're going to touch is probably like cadherens proteins. Okay, so there are cadherens proteins, and the Cry toxins have evolved a specific interaction encoded in the amino acids with these cadherens. So they can bind to the cadherens. Okay, that's the first step of the Cry toxin toxicity. Okay, and then there's a second protein. So this is step one. Step two is there's a second part of this where let's say you draw this. Let's zoom in on this thing right here. So this is a cell. Let's say it binds like a cadherin right here. Okay, here's the Cry toxin. So the first step is binding. The second step is in this cell membrane, there are transporters, like we talked about, and there's a big group of transporters called ABC transporters. ABC transporters, they do a bunch of different stuff. I think I got a picture of them on the... Here's the adherens junction. There's the cadherens. Here's an ABC transporter. These are the membranes, so it spans the membrane. So that means it gets stuck right in the membrane. It makes a little channel, okay? And then what these things can do, is they take stuff and insert it or bring it through or push it out of the cell. So the second step is these ABC transporters grab the Cry toxin and they bring it into the membrane. And then the Cry toxin opens up a big channel and causes this cell to die. So the Cry toxin is basically using the proteins that the midgut cells have against it, if that makes sense. It's evolved a way to grab the cadherin, and then the ABC transporter grabs the Cry toxin and brings it in, and then it kills the cell by opening up this channel. Okay, so that's how it works. So when you study the resistance of BT, like if you get an insect-resistant strain of BT, of a particular Cry toxin, most of the mutations are knockouts, or frame shifts. What's a frame shift? Yeah, so if you got the codons in the cell, each are made up of 3, and if you get say a deletion of like a single base, all of a sudden that whole frame will be shifted, and that will usually cause a premature stop codon to appear later in the thing. So usually it leads to a truncation of the protein. How would a knockout might happen? By a transposon inserting randomly into one of these genes. Okay? So what happens is in BT resistance, the insects literally like lose that cadherin, or they lose that ABC transporter. It becomes non-functional. Okay? Now you can imagine that's probably going to hurt the insect because that no longer has this ABC transporter or this cadherin in its midgut, right? That is going to cause a fitness cost. But that's going to be weighed against the selective killing of the BT, and it makes a choice. Is it better for me to not have this protein or die?
So they can actually survive without those.
Yeah, that's how you usually get the resistance. Because here's the thing. There isn't just one cadherin. If they only had one cadherin, maybe they would die. But they have like 9, maybe have 25 cadherins. And all these things are very, very specific to specific cell contexts. So maybe they have 5 different cadherins that are all important for midgut like linking. And maybe only one of these is the one that the Cry Toxin binds to. And so it just loses that one. And so you get a little bit of a loss of function, but it's something that it can deal with. These would be called paralogs. So if you have a bunch of paralogs of cadherins, which they do, this builds in redundancy and genetic diversity, which we talked about in the heuristics lecture, which makes them quote unquote anti-fragile. We talked about that in the heuristics lecture. So insects have a bunch of these things, and it helps them develop resistance to things like this Cry Toxin. The last form that you'll often see in resistance to BT are like midgut proteases. So what's this enzyme? Cuts proteins at what? No. You should memorize this because it's used in molecular biology and it's important in every single organism. Yeah, cuts at lysine and arginine. Cuts at positive. These are the positive. That's correct. Cuts at lysine and arginine amino acids. All organisms have trypsin as basically like a digestive enzyme. They just secrete it into their gut and it cuts proteins. Okay, so you can get mutations or knockouts in gut proteases. So remember how I told you how the BT is a crystal and part of its protein, the N terminus is responsible for the toxicity and the C terminus is a lot of times responsible for forming the crystals. And part of it becoming solubilized is it gets cleaved by gut proteases. And then this thing can now solubilize and become toxic. Do you remember that? Mm-hmm. Okay. So if you knock out the gut protease that does that cleavage, that can mean that there's less active Cry toxin in your gut, and therefore maybe you survive. So you also will see mutations in gut proteases. But very, very common to see ABC transporters cadherin mutations. And those are actually target site resistance. So that's why if you get resistance to Cry1A, which might be a particular enzyme that is good at binding lepidopteran cadherins, that doesn't necessarily mean you're going to be resistant to Cry2A or Cry2AB or whatever all these different things are, because it's target site specific. But there are some target sites that overlap. So that's that. This is good. We're good on time. Go to techniques to avoid resistance that the farmers use, pesticide people use, antibiotic people use. These are all kind of like general concepts, except the refuge strategy, which is kind of agriculture specific. So refuge strategy. We talked about this in the heuristics lecture. Or did we? No, we didn't. No, we didn't. There's a different example. Okay, so have you guys heard of the refuge strategy? No? Okay, so refuge strategy is, let's say you have field X. You make field X planted with BT cotton. Okay. Let's say year one passes, and 10 insects survive. All the progeny in the next year are going to be derived from these 10 insects that are resistant to your BT. That's a bad deal, right? Because then next year your BT completely doesn't work. Okay, refuge strategy is when you do this. You plant this big field, but in this part of the field, maybe, I don't know what percentage, there's people who mathematically calculate what percentage you want. Let's say 10% of the field you plant with susceptible cotton. What's that mean? It's not BT cotton, non-BT. Okay, now look at what happens. Maybe now this makes 5 insects survive over here, But over here, how many insects survive? Hundreds. Yeah, thousands, maybe even millions. Okay? But are they affecting your crop? No, not really, because if they come over here, they die. Okay? So you kind of like allow them to live in like a little portion of your field. And so then when they mate the next year, these millions reproduce, and some of these will reproduce, but it's not going to be many, right? It's not going to be many. So the refuge strategy is a way to slow resistance from developing. You can slow resistance of Bt from developing for like 20 years. It's still going to happen, but you can slow it. And if you alternate, if we think about, here's where the mathematics come in and population genetics come in, which I'm not a population geneticist. But imagine a scenario where these mutations in the Bt resistant strains are a fitness cost. Okay? They make that insect less fit. Now, if you have a refuge strategy, you can weigh the mathematics in a way where these ones are much more likely to survive than these few numbers because of that fitness cost. And that has to do with how big you make your refuge and like how big the cost is to the particular mutation. But it's a way to slow resistance. Does that conceptually make sense? That's the refuge strategy. It's a very, very, very famous strategy that's used everywhere. Okay, pyramid strategy. What's a pyramid strategy? Okay, it's like if you are, I'm trying to think of like a military example, like you want to kill somebody, but you want to stab them and shoot them and blow them up at the same time, and then they're really likely to die. It's kind of like that's what you think of with the insect. Like, so when they make these plants, these Bt plants, if you just put one cry gene, is it harder to evolve resistance to one cry gene or 3 cry genes? 3, because even if you evolve resistance to one, you're still going to get killed by the other 2. Okay, so it's a much less likely in probability scenario that you spontaneously get 3 mutations that give you resistance to 3 different cry genes. So the pyramid strategy is when they stack these different traits, and most of the Bt stuff is now evolving to this because you will get resistance automatically to a single gene. But if you make 3 genes, it's much, much, much harder to evolve resistance to that. Now here's the problem: is some people will plant fields, and over here they will plant standard generation 1 Bt, and then over here they will plant pyramid Bt. And this is a terrible idea because then you select for resistance here, and then they come in here, and then they can evolve resistance to the other mechanisms. And now all of a sudden you have a superbug, which is like the antibiotic superbugs that you hear of all the time. Okay? So that is a pyramid strategy. You hit them with more than one thing at the same time, and you basically kill them from multiple different targets. You can also monitor resistance. So that's, since we know that the ABC transporters and the cadherins are responsible for most of the endogenous wild Bt resistance, we can go and sequence genomes of fields. Usually what people do is on their field, if they have a Bt field and they see an insect, they'll take that insect, and you can send that insect in and you sequence the genome. And then we can look at and see what mutations are evolving. And if you see those mutation alleles popping up, maybe you want to increase your refuge, or maybe you want to crop rotate, use a different crop in the next year or something. I don't know. I'm not a farmer, but maybe that's what you want to do. You watch for it. High dose criterion. What's that? That's like have you ever got sick? They give you antibiotics and they say, do not stop taking these antibiotics until you've done the entire course. This is the same thing, because they want to give you a massive dose that kills 99.999% of all those bacteria. And you want to keep taking that, because if you take it in a low dose, you might only kill 98%, and that leaves 2% to survive that are now resistant. And then now you select for that. If you take a high dose, you're much more likely to kill everything. So this means that there's when they make the transgenic crops, they have to test them to see that they fulfill this. And there's EPA requirements that it has to kill 99.9% of all insects feeding on it. If it doesn't match that criterion, let's say it only kills 97% of the insects feeding on it, they will not give you permission to use that in the field, because that's just a mechanism that will select for resistance. Does that make sense? And then we can end with IPM. What's IPM? What? Really?
When you say it, I'm not able to.
IPM is integrated pest management. This is a big thing in entomology. So this means like when you're a farmer and you have crops, or if you're treating maybe even for insects or like mosquitoes, you don't just do one thing. You try to understand the whole biological system. And so maybe your insect pests, maybe there's certain points of the year where the temperature is ripe, where they're really likely to be really high at that level. So maybe that means you only need to treat with insecticide like right during those days, right? And it's not good to treat with insecticide before or after because you're not getting an effect and you're just selecting for resistance. Does that make sense? So having a broader understanding and also to like combining things. So BT, if you plant your field full of BT and maybe you start to see some resistance, maybe that's when you want to come in and use an insecticide to kill those things that might be evolving resistance. So maybe you're combining BT and insecticides, and maybe you got like a parasitoid that is also like attacking those, or ladybugs or something like that that's eating them. It's basically just learning a bunch of different techniques and trying to integrate them into a system that's specifically designed so that you don't encounter these problems like resistance. That's integrated pest management. We will end there.